Okay, so today I'm going to talk about the material covered in Chapter 18. I'm rearranging the lecture schedule a little bit because I think since we just talked about immunology, it makes sense to talk about the applications of immunology first before we go back and talk about pathogenesis and epidemiology. So today I'm going to tell you mostly about the application of immunology of vaccines, vaccination. Chapter 18 covers both vaccines and the use of antibodies or immunological factors like complement in diagnostic tests as well. But I'm not going to cover that part of the chapter. So really you just have to focus on the first half of the chapter where they're talking about vaccination. But in order to understand how vaccination works, we need to know how adaptive immunity can be acquired. So adaptive immunity is the kind of immunity that gives our bodies what for a pathogen, exposure to a pathogen? Claire? Specificity and memory. So memory is key for us for us to be immune. Okay, so either we're susceptible to a pathogen or we're immune. Once we've been exposed to that pathogen and developed adaptive immunity memory to that pathogen, then we are immune to that pathogen. But there are various ways in which we can be protected from infection by these pathogens. And one of them is active immunity, which gives us this long-term protection from the pathogen, or we can get temporary protection through passive immunity. And they're both natural means by which we can acquire active or passive immunity. Things like getting sick gives us active immunity, lifelong immunity to most pathogens. But there are also artificial means, such as vaccination, that will also give us this long-term protection from pathogens without having to suffer through the symptoms of the disease. And so I'm going to explain what a vaccine is. So it has to be part of a pathogen or an entire pathogen that is somehow changed so that rather than getting sick as you usually would being exposed to the pathogen, you get either no symptoms or very mild symptoms, but you get the advantage of this adaptive immunity, this long-term active adaptive immunity, the memory to the pathogen. The major trait that a vaccine has to have in order to induce immunity is what? What part of the, in general, what part is going to elicit an immune response? The antigens that are present, right. So antigens, so we don't need the entire pathogen because we can use just something that functions as an antigen. So that is the main difference between the two types of vaccines that are made, either attenuated, so the whole pathogen that's somehow been weakened, or inactivated, which can be a killed pathogen or part of a pathogen. And there are several features of vaccination or vaccines that are necessary in order to make them successful. So if a vaccine is going to cause more symptoms than just getting sick with the pathogen, obviously it's not going to be a successful vaccine. So there are a variety of factors, including how easy it is to store it, how expensive it is to produce, and how effective it is over the long term. So sometimes it's long-term but not lifelong protection, and so we have to have booster shots later in life. So, for example, your tetanus shot, you should get updated every 10 years. Otherwise, you can get sick with tetanus. The advantage of vaccination is that by providing this active immunity in a population, you are protecting the rest of the population that is not immunized from getting sick. Why? And this is only applicable to communicable diseases, ones that are passed from human to human and have no other mediator, like a vector, like a mosquito. Any idea? So what feature of vaccination, so let's say 90% of the population is being vaccinated against, or for example, the flu vaccine, right? We don't all get the flu vaccine. Maybe less than half the population gets the flu vaccine. But what does it do for the rest of us who don't get the flu vaccine? Right, so these, what Amy said was that the less people that get sick with or get infected by the pathogen, the less likely it is to be shed from those individuals and passed on to another susceptible host. So if the person next to you is vaccinated and they're exposed, they won't get the symptoms and bring them home and spread it to their roommates or their household members, because they won't have the symptoms. Their body will eliminate the pathogen before the symptoms that can lead to the spread of the virus. So this is particularly true of viral infections or bacterial infections that are communicable. So they don't have an intermediate host of an animal or a vector. And so herd immunity is that protection that we get even though we haven't been vaccinated. The other way that vaccines can help produce herd immunity is in the case of attenuated, which are still live pathogens, but they've been weakened in some form. In some cases, attenuated vaccines can be shed from the person who was vaccinated. So polio is a good example of this. It's an enteric virus that is shed in the feces, and so through vaccination of part of the population, that weakened virus can be shed from those vaccinated parts of the population to the unvaccinated parts of the population. They may acquire this weakened form of the virus, have develop the active immunity, the long-term protection, but not have those severe symptoms of the very virulent form of the polio virus. And you should know the key differences between these two forms of virus, other than one being still live pathogen and one being dead pathogen. There are also key differences as far as how we need to administer the vaccine, because they are two different forms. And there are many different forms of the inactivated vaccine. So inactivated can be the whole pathogen, or can be parts of it, polysaccharide, or even nucleic acid vaccines. It can be a recombinant vaccine, like we talked about yeast being used to produce the hepatitis B vaccine. And I'm going to go over the example of polio. Polio today is eradicated in the U.S. Since about 2000. No, before that. I think it was 1994 that it was declared as eradicated from the U.S., but it's still endemic in parts of the world. And we still, as children, continue to get the vaccine for polio. And that's because of world travel. There's always a chance that somebody getting sick from the virus abroad can bring it to the U.S., and so we still need protection. On the other hand, smallpox, we don't get vaccinated against anymore. Smallpox has been eradicated worldwide, so there's no chance of anybody traveling from somewhere else and bringing it to our country or our community. So there's no chance that we could get sick from smallpox anymore, unless you're actually working with the virus, which is still being used in research. And as far as the applications of immunology in diagnostic testing, I want you to understand that antibodies are key in their role. There are many different tests that depend on antibodies, either detection of antibodies, most often detection of antibodies in the patient's serum. So that portion of the blood that carries the antibodies. And so somebody that would have antibodies would be seropositive for antibodies to that pathogen. And there are many different ways that antibodies can be used to detect the presence of immunity in a patient. So, as I mentioned, immunity is adaptive immunity. I'm talking about this memory feature of adaptive immunity that's important for long-term protection against any pathogen. In the case of the active immunity, that's the long-term protection. So it results from either getting sick, your immune response when you're exposed naturally to the pathogen, the antigens present on the pathogen, or we can use artificially induce it using vaccination, which gives protection often for the community, even if they haven't been vaccinated. The other type of immunity is temporary. It's either early in life when the fetus is developing, there's IgG antibodies that are crossing the placenta, or you get IgA secreted from the mucous membranes in the breast tissue when the milk develops, and so IgA is in the milk. So this is the immunity of the mother that's passed to the child. It does not produce memory cells, memory BRT cells in the child, so it's only temporary. As long as they're nursing, they're getting protection. If the mother, let's say the family gets sick with the flu or a cold, the mother getting sick, she's going to have these antibodies circulating in her tissues, and she can pass it on to the infant through nursing. Or sometimes if we're having a hard time fighting off an infection, or if we have a particularly dangerous infection that may be as a result of toxin production by a microorganism, we'll get artificial protection that's temporary from the damaging effects of that pathogen or the products of the pathogen. So for example, tetanus. If you do get tetanus, the treatment is antibodies. Okay, so an injection of antibodies that are against the toxin. And that's really what does most of the damage during the infection is the tetanus toxin. So they can be produced, these antibodies can be produced in animals. Horse serum has been used, or from other people. Sometimes just to give protection to infants, an injection of gamma globulins, which is a mixture of antibodies, is given to help because the infant's immune system is not fully developed until they're about 6 to 12 months old. So these means, whether they're natural or artificial, only give temporary protection. After the antibody is cleared from the system, there are no cells that remain in the body that would recognize that pathogen upon subsequent exposure. So the child is still susceptible from getting that exact strain of the cold or strain of the flu or whatever that they've been protected from by their mother's milk during infancy. They could still get it later in life. So vaccination is basically a pathogen or part of a pathogen or a product of a pathogen, like a toxin that's secreted, that is able to induce active immunity, so this adaptive immune response that leads to long-term memory to that pathogen or to that antigen that belongs to that pathogen. And a successful vaccine needs to protect us from these life-threatening diseases. So we don't have a vaccine for everything. We don't have a vaccine for colds because they're generally not life-threatening. And there's so many of them that the chances of having a vaccine against one and then being exposed to some other one and still getting sick is more likely. So, but it does, vaccines do protect against life-threatening diseases, including measles, mumps, rubella, polio, smallpox. We also get seasonal vaccines against flu. And they give us a long-lasting protection. So it generates antibodies or memory B cells that can produce antibodies that would be specific for that virus or that bacteria. It has to be, in order to be successful, it has to be safe. So we can't have a lot of side effects that would be more damaging than getting the naturally infected with the pathogen. And it has to be very effective. Usually the most successful vaccines are 99% or more effective. Some of the vaccines we use have a little bit lower efficacy, around 90 or 95%. But something like a polio vaccine is 99% effective. Also, when you're thinking about, oh yes?
I have a question about the HPV vaccine and like what, because I haven't really kept like up with what was going on with them, so I was just wondering, like, is it actually work really, and like, cause you know it's newer, and is it doing anything, and is it really safe, and do you know if any like there have been like crazy side effects and stuff like that?
Okay, so the HPV is human papillomavirus, a vaccine that was introduced recently for teenagers who are sexually active because it's known to be, some strains of this virus are known to be connected with cervical cancer, which is a very severe, high mortality rate of cancer. So the idea was that to prevent the infection of the virus as a preventative measure for the cancer. And I'm not sure about the side effects. I haven't heard anything in the news about the side effects. It is recommended for sexually active girls to take it before they become sexually active, like around that teenage year. So your doctor would probably talk with your daughter at that time about taking it. But I don't think it's required. Like a lot of the childhood vaccines are required in order to, for example, attend school and such. Yeah,
Well, I was going to say, as of last week, FDC made official recommendation that all boys and girls get vaccinated.
boys and girls. Okay, so yes, boys and girls, because obviously the boys would be then passing it on to the girls during sexual intercourse. So having both vaccinated would provide this kind of herd immunity. If somebody didn't get vaccinated, they may still be protected. But no, I haven't heard anything about it not being effective, and it has to go through a lot of trials in order to get approved by the FDA. So far, has anyone else heard anything about inefficacy? So far, I've just heard that it's continued to be recommended.
I've only heard like rumors from people saying that that things, but I've never actually like seen in the news like actual like somebody actually got sick from this virus. I think it's just like people that are against getting vaccines or
Uh huh. Uh huh.
Well, I do have a question. So it protects against more than one strain?
I think there are a couple of strains that two major strains that they've chosen that are most likely to be associated with the cancer. There are many strains of human papillomavirus.
I got the vaccine, so I think it's 5 strains, 2
Uh-huh.
that are associated with it, and 3
That
are
aren't.
not.
Okay.
Yeah.
So 5.
So it's
Okay.
a combination of 5 inactivated viruses?
And you get it with 3 shots.
Yeah, I
over-
got it too. I'm so
Yeah.
happy.
So I looked into it.
Yep.
It's my downfall.
Yes.
I was told that like it's only like really effective in like because I didn't get the vaccine because like I choose abstinence, so like I don't like I was really pushed to kind of do it anyway because like what if like my future spouse like has HPV. HPV is the number one like STD going around, and so like my family was even like pushing it on me, like, what if you change your mind? But like I was like, I don't know. But like I was told that like it doesn't work whenever you're older. So like say if I get married at like 30 and then my husband has like HPV or like like that getting the vaccine like when I'm 30 wouldn't really work. But like so I should get it now just like, what I mean?
Well,
Like
it's not recommended to get it after age 26 for whatever reason. So it's only you like your doctor won't give it to you if you're over 26. So it's not that it's ineffective. I don't know. My doctor didn't.
I don't know. I'd have to look into that. It may be that in older individuals, they just get more severe symptoms of from the vaccination, or it may just be that they find it not to be effective in as far as by that time you've already been exposed to the virus, so it may not be protective at that point. But I'm not sure what the reason would be. I'd have to look it up. Yeah. Claire? Okay, so there are no passive vaccines. Vaccines are always active immunity, but they can be for like a long term or a lifelong term. Okay, so immunity. So what was the question again? Passive immunity is always temporary because your body will eventually clear those antibodies from the system, so they won't be there to protect you.
I think it's after 17, it says less than 6 months, right?
Yes, less than 6 months. Yeah, it would definitely be less than 6 months that they would maintain in your circulation. Any other questions about that? So the success of vaccination is demonstrated by smallpox, which is now eradicated worldwide due to a worldwide vaccination program. And polio is eradicated in the U.S., but it's still endemic in areas, especially areas of Africa and East Asia. There also, measles is now what used to be about a 1 in 200 chance of getting measles now is less than 1 in 1,000. And tetanus, also protective, but again, you have to have the booster. So if you wait more than 10 years to have the booster, there's a chance that you can get tetanus. And that's because it's not a attenuated vaccine, but it's an inactivated vaccine. So it's against the toxin, and it's an antitoxoid. So it needs booster. Often these inactivated vaccines need booster shots in order to give that longer term immunity. It tends to wane after a while. And we're always, research is always being done on developing new vaccines against the most life-threatening infections that are currently in the population. So currently efforts to develop vaccines against HIV, against malaria, against influenza. It's a yearly endeavor to develop vaccines against influenza, the seasonal flu, because of the rapid changes that occur from season to season due to the rearrangement, that antigenic shift, those big changes that occur in the strains of influenza virus in a season of flu infections. So I have a picture here of Edward Jenner, who is the gentleman in the red coat. He's associated or often called the father of immunology not because he was the first to practice vaccination, but really because he was the first to publish his ideas and results of vaccination. And he studied smallpox, and he was actually trained as a veterinarian, and he was traveling from farm to farm and heard the milkmaids saying, "Oh, well, I'll never have to worry about getting those ugly pock marks on my face because I got cowpox." And so he wondered about that connection between the virus that infects cows, that when people are in close contact with these domesticated animals, they can get the virus from the cows. So the milkmaids had actually gotten a much more mild infection, similar kind of symptoms with pock marks, these spots all over pus-filled spots all over your body. But he found that the milkmaids were no longer susceptible to the human form of the vaccinia virus, the smallpox. And he was not the first to use vaccination, but he developed it and published it, this practice of using the virus from these pus-filled lesions of the skin to inoculate susceptible individuals, in which case they would get a more mild version of smallpox. And so the death rate associated with this viral infection that's highly contagious was much lower as a result of this practice called variolation. So it wasn't actually vaccination, it was variolation, this practice of using the virus found in the pus of the pock marks to inoculate other individuals who hadn't yet been exposed. But since then, a vaccine was developed that was used not only in the US but worldwide. And the success of the vaccine is seen today in that, I think it was by 1979, smallpox had been eradicated from the US and most of the world. And I'm not sure of the date, but very shortly after that, maybe within the next 10 years, the vaccine was no longer used because there were no cases worldwide and because the chances of getting a severe infection with the vaccine was greater than actually getting getting smallpox naturally, getting the viral infection naturally. So now we don't get the smallpox vaccine for those reasons. So what are the two differences, main differences in the forms of vaccines? There are attenuated vaccines. This is a weakened form of the pathogen, so it's still a live pathogen. It's a microorganism or a virus that can use a host cell to continue to multiply and replicate. And so that's how it induces immunity. It just infects us, but it's a different form because it gives much more mild symptoms or no symptoms at all of that disease. Usually only needs a single dose to induce lifelong immunity because it's a live replicating pathogen. It can spread because it's alive. It can be shed from our bodies and spread to population that is not vaccinated. So this is what helps with the herd immunity. They will get the more mild form of the infection, but they'll get that long-term active immunity. Some examples of the attenuated vaccines include the measles, mumps, rubella, combination vaccine that we get as infants, one of the polio vaccines. It's the oral vaccine. It's no longer used since 2000. Instead, we get the inactivated form of the vaccine called the Salk polio vaccine, and also vaccines against yellow fever. Now, the disadvantages are it does replicate, so those individuals that are immunocompromised can get very severe symptoms, and that includes pregnant women, maybe the fetus if the pathogen crosses the placenta, and anyone who has an immunocompromising infection or is on some kind of medication that would weaken the immune system. So in general, inactivated vaccines are preferred because they're safer in this respect. They can't, they won't be more severe effects in immunocompromised individuals in the case of inactivated vaccines because the infectious agent is dead, unable to replicate, okay? Inactivated in the case of viruses so that it cannot use the host cell to replicate and make more copies. So it can't be shed and it can't produce more severe symptoms in an immunocompromised individual. So the key part that's maintained in the case of inactivated vaccines is the part that induces an immune response, the immunogen. And they fall into two basic categories. Either the entire pathogen is inactivated or killed. So the antigen, the complete set of antigens are present. Examples of this type of vaccine are the cholera vaccine, the vaccine for plague against Yersinia pestis, influenza virus, injected form. So now we have two types of flu virus vaccine. We have the nasal spray and we have the injection. But the injection is a whole inactivated virus. And the nasal spray is the attenuated virus. And then the Salk polio, as I mentioned, is also inactivated. So this is by injection that you get the polio vaccine. Fragments of the pathogen can also be used, and that includes the parts of the pathogen that make good antigens. So that would be what type of molecules? So proteins, well, the outer surface, but only parts of it. Yep, polysaccharides. Not particularly good are lipids or nucleic acids, although there are some nucleic acid vaccines that have been effective against things like West Nile virus. So it also includes things that are secreted, like toxoids. So these toxic molecules, antitoxins, our antibodies against the toxoids can be elicited by using these inactivated forms of the toxin. Okay, so they don't use the active toxin, but they somehow modify the toxin to make it what we call a toxoid, and it still functions as an antigen and elicits this immune response in our bodies. Hepatitis B is a recombinant vaccine against protein subunit or acellular vaccines include pertussis. Sometimes the capsule or the outer glycocalyx of the bacteria contains components that can function as antigens, and those are most likely polysaccharides. So pneumococcal vaccine is against a variety of different bacteria components of the capsules of species of bacteria that cause pneumonia. Now, the route of vaccination is important as well. Ideally, we want to introduce the vaccine just the same way that the pathogen would be introduced into the body, and so we can introduce the vaccine by oral methods. So the Sabin polio vaccine was just a drink. When I was a kid, we took it as a drink. We got, I think just once we took it, and then we got immunity. Now they're using the other form, the inactivated whole virus, and so in the case of inactivated vaccines, injection is the only way that you can administer them. But in the case of the attenuated vaccines, you can administer them orally, by injection, or nasally. So the advantage of this in the case of a pathogen like poliovirus, which infects the epithelial cells lining the gut, is that you get immunity to the virus right where it would find its host cell, so on the mucosa. But you can also get protection by injection, and that induces both humoral and cellular immunity, not mucosal immunity. And in the case of inactivated vaccines, especially when it's only part of the pathogen, often what you'll need is something to help it be better at eliciting an immune response. And those are additives that are combined with the actual antigen to make it more effective at mounting or eliciting an immune response. Those are called adjuvants. And also because the importance of vaccination early in life, because most of the life-threatening infections are more life-threatening in the younger population, 5 and under. So we get a lot of our vaccinations when we're very young. And so in order not to have so many injections, a lot of the vaccines are being combined now. So measles, mumps, rubella is 3 vaccines against 3 different pathogens in one shot. So the basic, this is a summary of the key differences between attenuated and inactivated vaccines. Attenuated are able to reproduce or multiply in the host. So as soon as you get vaccinated, it's going to reproduce and cause an infection, but ideally without symptoms or with very mild symptoms. So it's able to induce humoral immunity, so your memory B cells that would be producing IgG, also mucosal immunity in the case of IgA, and also cellular immunity, so T memory cells. The protection is very long-term, usually lifelong. There's no additional substances that need to be added in order to make it elicit a good immune response, so no adjuvant required. Usually only a single shot will do the trick for a lifetime. And there's the disadvantage can be that there is a chance that because it's replicating and reproducing, that the vaccine could change genetically into a more virulent form. And there have been cases of this, and there's a measured amount for each attenuated vaccine. There's a known rate of severe infections among the population of those people who get vaccinated. So, for example, 1 in about 200,000 people who get the polio vaccine will get poliomyelitis, so the most severe form of polio. Also, we don't want to administer this type of vaccine to immunocompromised or pregnant individuals because it is able to reproduce, and because of their immune status, they can get much more severe symptoms from the vaccine. And you have this flexibility of the method of administering the vaccine. You can do it by injection, orally, or nasally. But it does require a little more complicated means of storage, so often you need to store it refrigerated or frozen. And so that means it's going to be more expensive, and it's going to be more difficult to reach those individuals when you're thinking about worldwide schemes of vaccination, those individuals that live in very remote areas that don't have means of maintaining things at low temperatures for long periods of time. As far as the inactivated vaccines go, they're very safe because they're not able to reproduce. They're not able to mutate or change in any way because they don't reproduce. They're limited in the immunity they can provide because they only provide humoral immunity, so IgG, memory B cells only, not mucosal, not cellular immunity, or poor cellular immunity in some cases. They are short-term. Some of them can last tens of years, but often you need a booster shot in order to provide longer term immunity. And they often need a little help in eliciting an immune response, so an adjuvant or linking the antigen to a protein, so often like egg proteins like ovalbumin are used to link the antigen that's being used to elicit the immune response so that it provides a better, stronger immune response. So multiple doses might be needed because of this short-term protection that it might provide. But there's no risk. It's very safe, no risk to immunocompromised or pregnant individuals, and it's not as flexible as far as being administered as only by injection. But it's stable at room temperature, so long-term storage and transport to very remote areas is possible. So let's just look at poliovirus as an example. Poliovirus is among the smallest viruses, but even though it's a small virus, it can be quite virulent. In the most virulent form of the disease, it produces what's known as paralytic poliomyelitis. So it's an enteric virus. It infects our epithelial cells. And in most cases, 95% of people that get infected have no symptoms at all, clear the virus perfectly well, and then they have immunity. But in 5% of cases, it spreads from the epithelial cells of the gut to the muscle tissue, to the neurons. Okay, and so more severe symptoms are experienced, aches and pains and flu-like symptoms, all the way to temporary reversible paralysis. But in the very rare cases, this is 1 in 100 of, or less than 1 in 100, I think it's 1 in 200 of individuals who get infected with polio. It can spread to the neurons and cause permanent paralysis. Just some details about the virus. It's very small virus. There are 3 strains of the virus. Type 2 has been eradicated, but type 1 and type 3 are still endemic in parts of the world. And it has an RNA genome, which is a plus-stranded RNA. We talked about how that can serve as a template for protein synthesis and needs to make the minus strand for replication. It enters the cells intact by pinocytosis, and then it uncoats, releasing the genome, the RNA genome, for both transcription and for replication. And then mature virions are assembled, and upon exit, it lyses the cell. So in 1916, so this was an endemic virus in the U.S. And in 1916, there was an outbreak. And it was so severe and it spread so rapidly that there were actually, this is a sign from outside of a town in Pennsylvania, that the fear of the spread of the disease led to quarantining or of trying to avoid anyone infected to enter this town. They put up a sign. But in general at that time, pretty much everybody would be exposed to polio as a child. There weren't particularly good sanitation systems set up at that time, and so usually by the age of 5, everyone would have gotten exposed, and they would have had some partial protection, some passive protection from the earlier in life from their mother's milk, from the IgA antibodies there. After that time, let's see, at that time there were almost 30,000 people died, 30,000 people were paralyzed for life during that outbreak, and about 9,000 people died. And after that time, about 30 years later, in the 1940s, there was a scientist working on, and physician working on, developing protection to this infection. And his name was Jonas Salk, and he developed the first vaccine, which was an inactivated form of the vaccine. And he tested it and made it available to the general public, free of charge. And it was really through the efforts not of our government, because the CDC had not yet been developed, but through this Rotary Club network, that they raised all of the money that was used to administer the vaccine to school children And you can see here, this is a picture taken from, I think, Georgia, where they're all waiting for their vaccine. There are two lines, though. The black children are on one side and the white children are on the other side. But it was in this years of 40 and 50s that a second vaccine was developed by Dr. Savin, this one being an attenuated vaccine administered orally. So remember, the inactivated vaccine had to be injected. It had to be administered by trained individuals that knew how to use a syringe, so nurses and doctors. It gave humoral immunity, but not mucosal immunity. So the oral vaccine was what was giving protection from the infection at the site of entry at the gut. So the protection here in the inactivated vaccine was from paralytic poliomyelitis. But you could still get infected with the polio virus because it wasn't giving that mucosal immunity. So your epithelial cells in the gut could still be infected. Whereas the oral vaccine gave protection from infection of the epithelial cells, but it still, in rare cases, could result in paralytic poliomyelitis because the virus was still able to replicate, right? So it's an attenuated, not an inactivated form of vaccine. And so here are some comparisons of the advantages and disadvantages of the Salk vaccine, which is inactivated, versus the Savin vaccine, which is the whole virus but weakened form of the virus. And basically, since polio has been eradicated in the U.S., we just use the Salk vaccine because of its increased safety. And we no longer use the Savin vaccine, which in rare cases can produce paralytic poliomyelitis. And I think I'm going to finish there. We already had a little bit of discussion about the risks and the benefits, so I'll finish up with immunologic testing next class. If you didn't pick up your exam last class, you can come and pick it up now.